then anything that can't be disproven must at least be a possibility. I did find it rather interesting that some of the more purported, I don't want to say purported, but some of the more debated evidence we've seen was from as earliest as 30 years ago, according to what ChatGPT was able to dig up. So I am a little bit curious about what are the most recent comprehensive and scientifically conducted research projects that study the Loch Ness Monster phenomenon. Let's see what we can find out. All right, so it gave us, let's just scroll down real quickly. 5. Okay, that's a pretty good number. An environmental DNA survey led by Professor Neil Gamble, the University of Otago. Fieldwork in 2018, results public in 2019. A purpose-built systematic eDNA survey of Loch Ness, greater than 250 water samples across depths and seasons that screened for traces of vertebrate DNA and found no evidence for large extinct reptiles and detected abundant eel DNA, supporting the giant eel or large eel hypothesis as the most plausible biological explanation offered by that team. A basilisk. This is the most modern molecular and broadly scientific survey to date. Okay, yeah. And we sort of uncovered that in the first episode on Loch Ness, and it does seem to track that, the rumors of the plesiosaur, sure, that they're, if you base that on the sightings, it's sort of wishful thinking, I think, because, what's in there that the plesiosaur would be eating? Where is its food source coming from? Because it would have to have a large food source. So I think that was easily dismissed. But the large eel, giant eel, those really do track when you see some of the humps in the water. The local legends would have us believe. The BBC in 2003 sonar satellite survey, large acoustic sweep of the loch, a high resolution media-backed scientific sweep using hundreds of sonar beams plus satellite navigation to map and detect sizable targets. The survey had resolution sufficient to detect small buoys, but reported no animal of substantial size, which essentially means they found nothing. Operation Deep Scan, way back in 1987, coordinated. This is actually further back than in 1993, and here's Robert Ryan's expeditions right here. But let's take a look anyway. One of the most ambitious coordinated sonar surveys, 24 boats deploying echo sounders simultaneously, that swept the full length of the loch, produced several unexplained sonar contacts, but no conclusive biological identification. An important large-scale acoustic data set. Academy of Applied Science, that was what we covered in our previous episode 20. Long-term monitoring and field science by the Loch Ness Project. This is ongoing as I understand it. The Loch Ness Project coordinates repeated field sampling, sonar observations, and archive curation. It's less a single expedition and more a long-running program of field science, observation, data collection that documents sightings, sonar hits, biological sampling, and ground truth studies over decades. They have deployed robotic vehicles. They have surfaced historical equipment to film that feeds scientific reanalysis. So they're basically saying the most powerful one was this one right here at the top, the environmental DNA study. So it supports the large eel, giant eel hypothesis. Okay, that's an interesting term, or the way they phrase that, supporting it. I don't think it means they're saying we believe that this is a giant eel or a large eel that people are reportedly seeing. I believe what it's saying is you could rule out any animal that has a vertebrate or a bone system like the plesiosaur, but you can't eliminate the giant or large eel. All right, that's kind of where I fall on that. What say you? I'd interested to see what you guys think. Let's try. Why don't we try one more here? I want to take the eDNA and the BBC. Let's try this one. The BBC Sonar Satellite Survey projects. Determine what variables exist in those research projects that create or leave conditions under which the Loch Ness Monster could exist. And let's see if it's able to do that. All right, so it's going to compare the two projects. Looking for variables, blind spots, and methodological limits. Both studies reduced the plausibility of a large, easily detectable monster, but neither was logically exhaustive. Disproof, gaps in sampling design, detection thresholds, environmental processes, instrumentation, interpretation, and reference data leave limited possibilities where a large animal could be present yet undetected. Limited possibilities. So here's the variables when we're talking about the Loch Ness eDNA project. Spatial sampling and density placement. What is it? Where samples were taken. Surface versus depth. Near shore versus mid-loch. Across inflows and outflows. Okay, so why it matters? Because the eDNA was patchy. An organism that rarely visits sampled microhabitats or stays in deep confined areas could leave little to no DNA sampled locations. And given the rarity at which the Loch Ness is sighted, it would imply that this is a deep water animal that rarely makes appearances or does not go swimming around very often where it can be seen. Temporal sampling. What is it? How many times and what seasons times were samples collected? It matters because the concentration varies seasonally with behavior and migration breeding. Okay. A low frequency snapshot campaign can miss rare, transient, seasonal, present large animals. Okay. Do they migrate? From where are they migrating? I did hear a theory. I was unable to find it again that there are underwater estuaries that lead into the Loch Ness. Considering that a large animal would be able to fit in these underwater estuaries, that's an entirely different concept in my mind. Biomass, what is it? Amount of data an animal sheds into water per time, and the minimum concentration. Why it matters? A large animal that sheds DNA very slowly, or that it exists extremely low population density, may produce DNA below detection thresholds. Primer, marker bias and reference data. What is it? DNA meta barcoding relies on PCR primers and reference sequences to match reads on taxa. It matters because primers used don't amplify DNA from an unknown clade or amplify it poorly, or if an unknown animal's DNA is not present in the reference database. Okay, so it can't cross-reference anything if it has nothing to cross-reference it with. Contamination, yes, okay, this is going to be a huge one. Field lab contamination, it matters because stringent controls reduce false signals, but over-stringency inhibitors problems can suppress detection of rare DNA. So we can see that through both of these, here's the BB3 sonar, the sonar system's frequency, pulse length, beam geometry. Lower frequency long pulses are better for long range, but miss small or narrow targets. Okay. All right. We're starting to see, here we go. This is a big one. Depth, near surface blind spot. Many sonars have reduced sensitivity extremely near the surface and near the transducer, called the dead zone, and can produce poor resolution. A creature that spends most of its time just below the surface, creating surface signatures but little acoustic return, or very close to the transducer path, could be in a blind zone. So here's what we have. Again, and there's some similarities. The temporal snapshot versus behavioral avoidance. Okay, this is a big one because an animal that avoids boats, noise, mostly nocturnal, migrates, or is transient would be unlikely to appear during the survey window. So here's what we have. Variables where both projects can leave room for an undetected creature. Extremely low abundance. Yeah, one, maybe two individual creatures. Detection probability and sonar encounter probability decline rapidly for very rare animals. Behavior that minimizes detection, like we just read. It migrates. It shows up. It's at the surface. It's during night. It's during dark, so it can't be seen. Surveys separated, but cannot by themselves exclude an animal that arrived, left, or changed behavior between campaigns. Uncharacterized biology. And this one I find very interesting because this is what we mentioned a moment ago, that it's cross-referenced. A lot of these studies cross-reference data, but they can't cross-reference any data if they don't have any data to cross-reference it with. So they don't have a biological sample from which they can cross-reference to try and make a determination what, if anything, is down there. All right. So the final judgment, the project is the single most powerful, the DNA is the most powerful modern biological test of Loch Ness's fauna and strongly constrains many biological hypotheses. Basically, extremely low scenarios remain logically possible, though increasingly implausible. So you're telling me there's a chance. All right. A lot of good information here. We still are at a position where it's possible. Not likely, but it's possible. There is at least evidence to suggest that something's out there. We can't disprove it. And being a skeptic, as I mostly find myself, though I do wonder as I go on through producing this channel, how much more of a skeptic I will remain. If you're telling me you can't disprove it, then anything that can't be disproven must at least be a possibility. All right, that's it for now. Remember, as we go forward, tread carefully in those dark waters.